Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're all safe and well. Today, I'm delighted to be joined by Jeremy Robertson, who is the Global Healthcare Identification Solutions Manager, and Oya AKCELIK DURMUS from Central Europe Marketing Manager for Health and Personal Care, and they're both on bioMerieux. And today, we're going to be talking about microbiological identification within pharma. So, Oya, Jeremy, lovely to see you both.
Great to be here. Thank you,
Yeah,
Rizwan.
good to meet you, Lisa. Hello.
Great. Well, look, before we start talking about microbiological identification, perhaps you guys could just give people a bit of a background on your roles within bioMerieux.
Yeah, sure, with pleasure. I'm working for 14 years in bioMerieux, and since the beginning, I'm working for the industry division, meaning with pharmaceutical and food industry. And since 3 years, I'm based in Germany and managing healthcare marketing in Central Europe cluster, working closely with pharma industry to help them with their microbiological quality control.
Fantastic. And Jeremy?
Yes, I'm on our global marketing team. I'm responsible for our identification ranges, from our manual ID ranges, such as our API strips, to our automated ID ranges, such as Vitek. So thank you for today. Great to talk with you.
Yeah, it's lovely to meet you both, as I said. So before we start talking, obviously, about microbiological identification, perhaps one of you could give me a bit of a background on bioMerieux for those people not familiar with the company.
Yes, well, it goes back to 1 century, even more. bioMerieux story is about people who share the same vision and passion to serve public health. And bioMerieux is a family-owned company created in 1963. But this human and scientific adventure began more than 57 years ago. Its roots reach back to tradition of Louis Pasteur and to fight against infectious diseases. In 1897, Marcel Merieux, who had studied with Louis Pasteur, founded a laboratory in Lyon where he first developed his first anti-tetanus sera. And things moved even faster under his son, Dr. Charles Merieux, and grandson, Alain Merieux. They founded gradually buildings and laboratories to develop vaccine for human and animal health. So Institut Merieux grew to become a world leader in human and veterinary vaccines. In 1994, Alain Merieux shifted his focus from vaccine business to diagnostics to develop more effective clinical and industrial tests to protect patients and consumer health. And adventure still continues, innovative and independent, still based in Marseille-l'Étoile with multidisciplinary research collaborations. Now, bioMerieux has more than 10,000 employees worldwide, existing directly in 44 countries with 20 research and production sites.
Brilliant. Well, thank you for giving that lovely history lesson on bioMerieux. I'm sure a lot of people learned a lot of things that they didn't know about before. Now, let's talk about COVID-19. You mentioned vaccines there in the history of the company.
Yes.
So how has COVID-19 impacted bioMerieux as a business, both in terms of yourselves and how you serve and support your customers?
I think Jeremy can also agree. For sure, it was difficult times as human being for all of us. We needed to arrange ourselves to that new normal, having in mind that our job is more and more important as we are a key player in public health. Luckily, as our company had already some solutions for remote working, remote support, we had a rather smooth transition during the pandemic onset. We have organized ourselves to digital working and communication to be able to manage the constant supply. So our teams are working daily with our partners, customers to determine the most urgent needs. And during this restricted time, bioMerieux remained highly engaged to help healthcare professionals to fight with COVID-19 pandemics with the urgent development of 3 complementary PCR tests. What matters the most is in these times is providing them rapid and reliable tests, so you can be assured of our total commitment.
Right. And Jeremy, have you got anything to add? Is there any difference in terms of how you've been working in North America compared to Europe?
Yeah, I think Oya covered it. I would just add that, it's certainly been challenging. The fact that we're a global company means that we have supply chains spread around the world, which has presented its own challenges, but also some benefits, I would say. It's allowed us to make changes and shifts, which has allowed us to continue serving our customers faithfully throughout the pandemic. So it's been very impressive to see, I think.
Fantastic. Well, look, one more question related to COVID. before we talk about identification. Obviously, everyone's searching for a vaccine. We had the great news, obviously, from Pfizer earlier this week. So how do you at bioMerieux, how can you help researchers and scientists in terms of finding and developing a vaccine quickly?
Yeah, vaccines are generally quite huge molecules that are often complicated to produce, and therefore one batch worth a lot of money, sometimes millions. Vaccines also cannot be just sterilized like small molecules, so it is even more critical to monitor microbial contamination during the whole process of production. As humans are the most common sources of contamination, automatic in-process control is getting more important here. To improve microbial control, rapid microbial detection like rapid sterility testing and therefore fast microbial identification is of the essence. Vital information can be obtained for the sake of final product in the end by identifying the contaminants fast. So bioMerieux here provides a wide range of phenotypic identification methods ranging from API, an easy and cost-effective method for small producers, and then VITEK to compact, a closed, fully automated system for mid-sized producers, and finally VITEK MS, multi-top mass spectrometry for big producers with high throughput. My dear colleague Jeremy will mention about that. And here the important thing that we do is giving pharmaceutical and vaccine producers the power to protect in the end patient and consumer health by providing them fast and reliable information about identification.
Right. And Jeremy, have you got anything to add to what Oya just said there?
Yeah, nothing really more to add except to say that I think, you know, Oya talked earlier about the history of the company and our strong roots in microbiology. And I would just add that, you know, that really influences everything we do. It's something we're very proud of. So it's really a big part of the company, and it's something that we take very seriously. And it drives what we do and how we build and make our products and how we serve our customers. So,
right, brilliant. Yeah. Right, so let's talk about microbiological identification. So, what changes have you seen in the sector, and what technology is actually available to researchers and scientists out there?
You know, I think as many of your viewers can appreciate, Rizwan, the field of pharmaceutical microbiology can be a very conservative field where often change can take time. Yeah. Sometimes techniques from our grandfather's time are still in use. I think for several years now, there has been a strong trend to eliminate human error. I would say today more than ever, this trend is in focus as regulatory authorities have an eye on data integrity, placing more and more importance on the software and IT infrastructure of ID solutions. And I see vendors responding with more advanced software options that better address the needs of pharmaceutical manufacturers in this area.
Right. And Oya, would you have anything to add to that?
Yeah, the feedbacks that our pharmaceutical customers provide us here is very important, as Jeremy said. We shape the improvement in such a way.
Right, okay. So can one of you tell us a little bit about the history and evolution of microbiological identification in respect to API and VITEK?
Yes, actually the determination, identification of bacteria based on various characteristics is one of the flowstones of microbiology. It started with petri dishes, which are fed with various culture media, and these culture media contain reagents and indicators for the detection of certain properties that allow the determination and discrimination of bacteria at the level of genus or even species. But you got to do a lot of plates and tubes each time when you want to make an identification, so it's not an easy job. To streamline the process over time, petri dishes were exchanged by reagent tubes and finally small wells, small open wells on a strip. And this was actually in that time the automation, and it was the start of the API systems. Initially developed in France, commercialized in the 50s, then it's transferred to US. Then in 1986, bioMerieux acquired the API systems, and still it's in Laban. And the next step in the evolution was to transfer this open wells to miniaturized closed carts. The expert area of my dear colleague Jeremy, it was the start of VITEK range. Vitek is a combination of the terms in vitro and technology, and has been invented by actually spacecraft company McDonnell Douglas. Initially, it was developed for the use in space, and in '60s, it was the NASA space program was looking for an automated device to monitor microorganisms in space because astronauts were acquiring infections. And as we cannot send a doctor with them in that time, an automatic identification and which recommends the correct antibiotic treatment from a distance was needed. So Vitek was born, and it became very famous. Then in 1988, bioMerieux acquired this system and improved ever since. And in 2005, Vitek 2 Compact was launched just for the pharmaceutical industry needs.
Right, well, that's really interesting. It's amazing how a lot of this technology used with NASA has been transferred over now to more day-to-day use as well.
Yeah.
So let's talk about the importance of microbial identification in pharma manufacturing then. Why is it important?
Rizwan, I think the importance of bioID is really closely the product that pharmaceutical companies are manufacturing. If they find issues with microbes in their product or in their manufacturing environments, they need to know information about those organisms in order to mitigate the risk of product quality issues and ultimately patient health. With over 50 years of experience in the microbiological diagnostics field, I understand very well how important the quality and accuracy of the results from our tests are to our customers, because they're using these results to make decisions that impact the quality of their products. And this is why we place such a high priority on our internal quality standards. I think a really great example of this is how we build our knowledge base for our ID systems. Our quality standards dictate that we incorporate both microbial and technical variability into our knowledge base. We do this by collecting multiple strains using different parameters for each organism that we add. Parameters like geography, source, operator, growth conditions, and several others allow us to build a very robust knowledge base that includes both interspecies diversity as well as variable test conditions, so that our customers can rely on their Vitek system for accurate identification and hence make very confident decisions about their product quality.
Right. Okay. Oya, have you got anything to add to that?
Yeah, it's very, the system is open to improvements, even from distance each year.
Brilliant. And so what do you see as the main challenge in terms of microbiological identification? And how do you as a business help people address those challenges?
I think a really big challenge of any ID method is the ability to resolve an organism's identification to the level that's required by the customer. Different ID technologies have different limitations with respect to group, genus, species, or strain level ID. And the criticality of the sample usually determines the desired level of resolution. Typically, more critical samples, such as those from a contamination investigation, will need a higher resolution ID to the species or maybe even the strain level. And often customers will employ different molecular techniques such as 16S gene sequencing or multi-locus sequence typing for these type of critical samples to get the desired level of resolution. But molecular methods can be costly and time consuming, so many customers will utilize faster and more convenient traditional phenotypic methods and even newer proteotypic methods for day-to-day routine samples. And these methods can provide a species level ID in the majority of cases, and that's usually good enough for routine ID samples. Another challenge I see increasingly is the need for mold identification in pharmaceutical quality control environments. And traditional methods for identifying molds are highly subjective, very labor intensive, and time consuming. Molecular sequencing methods can be less subjective and offer better resolution to species level, but many QC labs don't have these capabilities, so they have to send isolates to third-party reference labs for molecular mold ID. And as I was just referencing, this can be very costly and time consuming, can take them several days or sometimes even weeks to get results back. So our Vitek MS system uses MALDI-TOF proteotypic ID technology to provide microbial IDs in minutes. And our robust knowledge base includes a variety of more than 200 fungal species. So it's a really great option for a quick and accurate mold identification to the genus or species level.
Great. And Oya, have you got anything that you'd like to add? Any other challenges Jeremy's missed out that you want to add?
Yeah, I totally agree. Here, the priorities of the lab is very important, as Jeremy said. So this was what I would like to mention.
Right. Okay. So looking ahead, what further developments do you see on the horizon in terms of improving microbiology? I have to say this a lot of times to get it right. Microbiological identification. So what are the sort of future developments you see within that area?
You know, I think continued development of the knowledge base is really critical to any commercially available ID system. And no ID system on the market today can provide 100% coverage of all microbes, especially environmental species, which are what pharmaceutical manufacturers encounter most frequently. And as we know, there are far more unknown bacterial species in the world than currently characterized. So this is why we continue to offer our customers taxonomy changes and new species claims through regular knowledge base updates so they can benefit from the latest developments in the field of industrial microbiology. Another development I think you'll see in the future is a more holistic offering from identification system providers. We recognize that our systems are not just a box that's sold, plugged in, and ready to go. From the evaluation phase to the qualification phase to the implementation phase, pharmaceutical QC labs have to ensure proper documentation and validation,
Sure.
and they often need guidance in these efforts. At bioMerieux, we've developed a comprehensive ID solution for our customers that includes not only the ID system itself, but also support guidance needed from evaluation right through to implementation. For example, we provide our customers a user requirement specification package to help them prior to acquiring the system during their evaluation phase. And then post-acquisition, we offer full training and even validation documentation to help get them up and running quickly so they can start benefiting from their new ID system as soon as possible. I think we all recognize that bringing a new analytical device into a pharma lab can be a real challenge. I think you'll see more of this type of support from vendors because it helps make that process quicker and quicker. And I think it's really much needed in the industry.
Brilliant. Well, thank you for giving me such a great overview of microbiological identification. I mean, it's a really interesting topic, actually, and it is of the moment as well, given what's going on in terms of the search of COVID as well. How can people find more information if they want to sort of get more information about this particular subject? Where can they turn to?
bioMerieux is on LinkedIn, YouTube, Twitter, or you can just type on internet bioMerieux, and there is the www.biomerieuxindustry.com website that was developed especially for industry customer needs. There one can find different kind of information like on-demand webinars, posters, and other solution-specific informations. And we have our digital technical library available on www.mybiomerieux.com. So all of our customers can register for free, download the bioMerieux package inserts, certificate analysis, or user menus. And we are also very happy to exchange in person. If nowadays this is not possible, you can call us anytime or reach us via digital channels.
Fantastic. Well, all that's left to say is, Oya, Jeremy, thank you very much for your time. That was really interesting. I appreciate taking time out to go through that with me. So thank you.
Thank you.
Thank you as well for the opportunity.
Great. And thank you everybody for watching. I hope you found that interesting. As Oya and Jeremy said, if you'd like to know more about microbiological identification, then check out the links which will be above the video, where you can find out more about this particular topic. And if you want to speak to Oya, Jeremy, or have any questions for them, feel free to leave them below the comment section or message them on LinkedIn, because they will be tagged on the post, so you can message them directly if you want to do that as well. So all that's left to say is thank you very much, Oya and Jeremy, for your time. It was lovely to see you.
Thank you
Great.
as well.
Thank you.
And thank you all for watching. And as always, until next time, stay well and stay safe. Bye-bye.